my name is Jessica Cook, and I'm a fourth year DDS PhD student at UCSF in the lab of Ophir Klein. Today I'll be discussing my project, which is entitled Unraveling Wound Healing in the Oral Mucosa. Distinct subpopulations of fibroblasts exist within the tissue to facilitate wound repair. The oral cavity consists of hard tissues and soft tissues. The soft tissue within the mouth is referred to as the oral mucosa. Morphologically, this tissue resembles the skin. However, because this tissue exists within a unique environment that involves constant stress as a result of repetitive damage from mastication, as well as signals from allergens, foods, and microbes, it has developed unique properties when compared to the skin. One of which is its ability to quickly and efficiently heal wounds without a scar. This is seen here in a study involving matched dermal and oral wounds in humans. You can see that the oral wounds heal much quicker and typically without a scar. Despite this intriguing phenomenon, very little is known about the cellular and molecular dynamics of oral mucosal wound healing. My project specifically focuses on fibroblasts, support cells that exist within the tissue. Fibroblasts are, in general, responsible for laying down extracellular matrix to plug the new wound bed, signaling to epithelial and immune cells to stimulate proliferation, migration, and cytokine secretion, and reorganizing the provisional extracellular matrix to resemble the unwounded tissue architecture. But little is known about the subpopulations of fibroblasts that exist within the oral mucosa and how they contribute specifically to regeneration. Therefore, my project is broken down into three aims. Aim 1 is to characterize the identities and functions of these subpopulations. Aim 2 is to determine the temporal response to wounding within the oral mucosa. And Aim 3 is to define the signaling pathways utilized by OMFs to facilitate regeneration. To answer these questions, I used single-cell RNA sequencing of matched samples from the skin and oral mucosa of mice. The samples were collected day 7, day 4, day 2, and day 1 after wounding. Pictured here is a 2D projection of the 15 cell populations identified within the dataset, 8 of which are unique OMF subpopulations. Clusters were identified using gene expression captured by the sequencing. In addition to identifying these subpopulations, I was able to determine the temporal distribution of each cell type and assign tentative functions. Next, using network analysis, I was able to group the fibroblast subpopulations based on their signaling patterns and determine the signaling pathways most utilized by each fibroblast subpopulation. Altogether, my work allows me to generate a working theory of OMF cellular and molecular dynamics in wound healing, involving a trade-off between OMF subpopulation ratios and signaling pathway utilization. My future work involves validating my analysis in vivo. Thank you.